Hi guys, in this video we'll be talking about recombinant DNA. The formation of a recombinant DNA can be created in the laboratory. A restriction endonuclease, which is an enzyme, is selected to produce cut DNA fragments at specific points, leaving them with sticky ends. The two cut DNA fragments are then incubated with another enzyme known as DNA ligase. DNA ligase is responsible for sealing the cuts in the DNA and for the formation of covalent bonds between the DNA fragments. These recombinant DNA molecules can then be introduced into bacterial cells to produce clones. Now let's talk about gene cloning in bacteria. Gene cloning is the process of manipulating DNA to produce many identical copies of a gene or another segment of DNA in foreign cells. The following steps are involved. A recombinant DNA, as I mentioned earlier, is produced, and a plasmid which serves as a vector is prepared. The plasmid must carry a gene that makes bacteria resistant to antibiotics, in this case ampicillin. The DNA to be cloned often interrupts the LAC-Z gene, which is a gene that codes for an enzyme that breaks down galactose. The LAC-Z gene and the ampicillin resistance gene are called selectable markers, and they are important for researchers to search which bacteria in the colony contains the recombinant DNA, because some bacteria do not have the recombinant DNA inserted into them. The reaction mixture for producing the recombinant DNA is introduced into the bacteria, and the foreign DNA is taken up the bacteria through a process called transformation. This then produces a recombinant bacteria. The bacterial cells are then applied to a petri dish that is supplemented with ampicillin and a derivative of galactose X-gal, which causes the bacteria to turn blue when it is broken down by the enzyme that is coded by LAC-Z gene. The bacterial colonies that contain the recombinant DNA are now identified. This is because the bacteria that grow on petri dishes with ampicillin contains either recombinant DNA or only plasmid. The blue bacterial colonies contain LAC-Z gene, which means that no gene is inserted into them, which means that the ones that are white in colors, that's the white colonies, are the ones with the recombinant DNA. The cells with the recombinant DNA, which are the ones colored white, are then selected and grown to produce a large population. The recombinant DNA are then isolated and purified from the bacterial cells, and further analysis techniques are used to confirm that the correct recombinant DNA has been